Assalamualaikum. My name is Ma'ina Nadla. I'm from DBT4A. I will be performing practical test which is sterilization technique. Sterilization, which is any process, physical or chemical that destroys all forms of life, is used especially to destroy microorganisms, spores and viruses. Sterilization can be achieved through various means including heat, chemicals, irradiation, high pressure and filtration. Sterilization is distinct from disinfection, sanitization, and pasteurization, in that those methods reduce rather than eliminate all forms of life and biological agents present. Tissue culture is in vitro cultivation of plant cell or tissue under aseptic and controlled environment conditions, in liquid or semi-solid. The whole process requires a well-equipped culture laboratory and nutrient medium. This process involves various steps via preparation of nutrient medium containing inorganic and organic salts, supplemented with vitamins, plant growth hormone and amino acid, as well as sterilization of explants, glassware and other accessories, inoculation and incubation. In this practical test, I will perform a few types of sterilization which are heat, air, UV, filtration and chemical sterilization. Firstly, we start with the heat sterilization that consists of two, which is dry heat and moist heat. The first one is dry heat. Dry heat sterilization techniques requires a longer exposure time, which is 1.5 hours to 3 hours, and higher temperatures than moist heat sterilization. Various available methods of dry heat sterilization are hot air oven, incineration, and flaming, which is scalpel and wire loop. For example, I use kitchen knife as replacement of scalpel that being sterilized bypass through the flame using candle that have a same function as Bunsen burner. Next is moist heat. Moist heat sterilization is used to heated high pressure steam to sterilize the object. Moist heat has better penetrating power than dry heat and at a given temperature produce a faster reduction in the number of living organisms. Steam sterilization is non-toxic, inexpensive, rapidly microbicidal and sporicidal. Moist heat sterilization using autoclave is commonly used for the sterilization of biohazardous trash, heat and moisture resistant materials such as aqueous preparation. Next is air and UV sterilization by using laminar air flow. It is a carefully enclosed bench designed to prevent contamination of biological samples or any particle-sensitive material. Laminar air flow does sterilization by using ultraviolet light. Air is drawn through HEPA filter and blown in a very smooth laminar air flow toward the user. Next is filtration sterilization. Filtration is an effective and reasonably economical method of sterilization. It is used to sterilize heat-sensitive fluids and air. It is particularly useful for solutions containing toxin, enzymes, drugs, serum, and sugar. Sugar solutions used for the cultivation of microorganisms tends to caramelize during autoclaving, and so they are best sterilized by filtration. Filters with non-pore size which are sufficiently small to hold back bacteria employed. Shrink filter is a single-use filter cartridge. It is attached to the end of a shrink for use. Shrink filters may have lever lock fittings. The use of a needle is optional. Where desired, it may be fitted to the end of the shrink filter. A shrink filter generally consists of a plastic housing with a membrane that serves as a filter. The fluid to be purified may be cleaned by drawing it up the shrink through the filter or by forcing the unfiltered fluid through the filter. Shrink filters may be used to remove particles from a sample prior to analysis by HPLC or other techniques involving expensive instruments. Shrink filters are quite suitable for slang line work, which makes extensive use of needle and shrink. Being relatively affordable, they may be used for general purpose filtration, especially of smaller volumes where losses by soaking up filter paper are significant. Shrink filters are also available for the filtration of gases and for the removal of bacteria from samples. Lastly is chemical sterilization. Chemical sterilization is the process of removal of microorganisms by the use of chemical bactericidal agents. Even if physical methods of sterilization are more appropriate for effective sterilization, it is not always appropriate to use for heat-sensitive material like plastic, fiber optics, and biological specimens. Under such conditions, chemical either in liquid or gaseous state can be used for sterilization. However, it is critical to ensure that the material undergoing sterilization are compatible with the chemical being used. Besides, it is important to adopt safety rules in the workplace safety during the use of chemical agent. The chemical method of sterilization can be categorized as liquid and gaseous sterilization. For example, spray the working area with 70% of alcohol and wipe it with a tissue. Alcohol kills germs using a simple chemical process called denaturation. Denaturation occurs when alcohol molecules degrade the protein formed in germ structure.